Good morning, good afternoon, and good evening, everyone. This is Liam Sanyo from Inside Scientific, and I'll be your host for today's event, which is titled Cellular Mechanisms Behind Alcohol Abuse and Binge Drinking Behavior. This webinar has been sponsored by HICA, a division of Harvard Bioscience. Today, we're very fortunate to be joined by Dr. Gilles Martin, Associate Professor of Neurobiology at the University of Massachusetts Medical School. Today, Dr. Martin will discuss the cellular basis and reward pathways behind binge drinking behavior and alcohol abuse in rodent models. And with that, I'm very pleased to welcome Dr. Gilles Martin. Thanks so much for joining us today, Gilles, and feel free to take it away whenever you're ready.
Thank you, Liam, and good morning, and thank you all for coming to this webinar. I also want to thank Harvard Bioscience and Inside Scientific for the opportunity to share my research today. I will be talking about the cellular mechanisms. Okay, let me just move this. Cellular mechanisms underlying the effects of binge alcohol drinking on synaptic transmission in the nucleus accumbens, and more generally, how the accumbens microcircuitry adapts to repeated alcohol consumption. So my research focuses on binge drinking, a stage of the addiction cycle that affects primarily late adolescents and young adults, an age when the brain is still very much developing and as such is particularly vulnerable to alcohol. Also, binge drinking is a well-recognized gateway to dependence. So nobody will be surprised if I say that alcohol addiction is a serious problem in the US as it is in many other countries around the world. However, because it is one of the few legal drugs with nicotine and now cannabinoids, at least in some states in the US, that is readily available, it is easy to underestimate the full magnitude of the problem. So to illustrate this point, I would like to share actually a couple of very, very short stories. So the first is 2 years ago, I was discussing a paper on alcohol addiction with a bunch of graduate students here at the University of Massachusetts Medical School. And the first thing I did was to ask them to raise their hands if they knew someone, a friend or family member suffering or struggling with alcohol addiction. And to their surprise, but not really to mine, nearly half of them really did so. So alcohol addiction and binge drinking. Well, there is a survey that came out a couple of years ago that I found on the website of the National Institute of Alcohol Abuse and Addiction that really shows the extent of the problem. About 7 million teens between the age of 12 and 20 drink monthly, and nearly 1 million young people reported binge drinking on 5 or more days over the past month. More generally speaking, 9 million adults are considered alcoholics, and the cost on society is quite large indeed, estimated between $40 and $60 billion due to a loss of production, healthcare-related issues, and crime. Also, alcohol is responsible for 100,000 deaths annually. But maybe one of the most sobering facts is that there is really no specific treatment, no good treatment against alcohol addiction. So the nucleus accumbens as an addiction hub. The nucleus accumbens, here shown in green, is part of the mesocortical limbic pathway that originates in the ventral tegmental area, shown in blue. The ventral tegmental area extends dopaminergic projections to the nucleus accumbens, but also to other forebrain regions like the prefrontal cortex in red and the amygdala in yellow, but also the hippocampus. So here is a coronal section of the brain showing the dorsal striatum, and ventral to it is the nucleus accumbens that is typically divided between the core and the shell region. The core is anatomically associated with the dorsal striatum. In contrast, the shell is typically associated with the extended amygdala. So in addition to the dopaminergic projections that I just mentioned that are coming from the ventral tegmental area, the main feature of this brain region is the fact that they receive massive glutamatergic input from a number of brain regions. And here I've listed probably the 4 main regions. These regions are the prefrontal cortex, the thalamus, the amygdala, the hippocampus. All the data I'm going to show today have been collected from the core subregions, so I'm going to, for now, to get rid of the inputs that really synapse on the shell part of the accumbens. But the point I really want to drive home today is the fact that these brain regions really send extremely different kind of information to the nucleus accumbens. As you know, the prefrontal cortex is associated with executive function, thalamus with sensorial, amygdala with the emotional information, and finally the hippocampus with contextual information. So how the accumbens really computes and integrates this sometimes contradictory information, it's something that we'll see in a couple of minutes. So here on the left is a cartoon of a very simplified version of the microcircuitry of the nucleus accumbens. Those are the main players with the medium spiny neurons at the bottom in green. And they come, and I should really add that the medium spiny neurons are the only output of the nucleus accumbens. So whatever really changes the excitability of these neurons will likely have an impact downstream on the behavior. So these medium spiny neurons typically come in two flavors, those expressing dopamine D1 and those expressing dopamine D2 receptors. In addition to these medium spiny neurons, we have GABA interneurons and finally the cholinergic interneuron on top in gray. And in addition to that, as we saw in the previous slide, there is the dopaminergic inputs and the afferents, glutamatergic afferents from the cortex, the BLA, hippocampus, and thalamus. But because I'm not really going to talk about GABAergic synaptic transmission here, I just decided to get rid of that. And so today I'm going really to talk about the three projects we have going on in the lab. And these projects are different states of advancement. The first one has to do with the cholinergic interneurons and their role in regulating glutamatergic synaptic transmission in MSN. The second one in green really focuses very specifically on glutamatergic synaptic transmission coming from the cortex and the BLA. And finally, the third one is about neuronal ensembles, also called engrams, in relation to binge alcohol drinking. So let's start with the first project. The role of cholinergic interneurons on glutamatergic synaptic transmission in the nucleus accumbens. Why should we really care about cholinergic synaptic transmission and alcohol? Well, from an epidemiological standpoint, we know that, there is really a large number of adults, 35%, who consume alcohol and that also smoke. And more than 6 million Americans, for instance, suffer from dual alcohol and nicotine dependence. Functionally, we also know that acute alcohol really does change the excitability of cholinergic interneuron, which results in an increase of the release of acetylcholine in the nucleus accumbens. However, there are really a number of outstanding questions. For instance, do cholinergic interneurons modulate MSN excitability directly, indirectly? What are the cellular mechanisms? Is this modulation sensitive to binge drinking? And finally, do cholinergic interneurons influence drinking consumption? So again, let's go back to this little cartoon. We have our main players, the medium spiny neurons, the cholinergic interneuron CIN, and the dopaminergic input and glutamatergic inputs. But before we can answer these questions, we have a couple of technical challenges that we have to overcome. First, we need to be able to control very specifically the excitability of cholinergic interneurons. Well, there is really a small problem with that, is that cholinergic interneurons represent less than 1% of the total neuronal population of the nucleus accumbens. So it's literally looking for the needle in a haystack. So how do we really achieve this level of control? Well, for that, we turn to a number of transgenic mice models. The first one is the chat channelrhodopsin. So this mouse model is, in this mouse model, the channelrhodopsin is constitutively expressed very specifically in cholinergic interneurons because the expression is driven by chat. And for those who are not familiar with optogenetics, channelrhodopsin, here is CHR2, It's a protein that is light sensitive and that changes conformation when excited by blue light, allowing an influx of sodium into the cell and triggering the excitation of the cell. And here, what you are seeing is a typical cholinergic interneuron that is fluorescent, that is expressing channelrhodopsin. When you turn on for a very brief period of time, 1 millisecond here, the blue light, you can see that this neuron fires very regularly based on the stimulation pattern that you chose. But what we also want to do is to inhibit these neurons. So how do we do that? We use a very, a different mouse, which is a Chat-Cree mouse. In this particular mouse, what we do is that we inject viral vector, which is AAV, double flux with, in this particular case, archaeorhodopsin. Archaeorhodopsin is also, it's also a light-sensitive protein, but this time, when it is activated by light, it will inhibit the cell, as shown on the graph on the right. You can see that when the light is turned on, the spontaneous activity of cholinergic interneurons is totally interrupted. The second issue that we have is to be able to really differentiate between the medium spiny neurons that express D1 versus D2 dopamine receptors. So how we do that? Well, this is where we combine the transgenic mice that I just described with the dopamine D1 receptor TD tomato mice. On the left, you see what an electrophysiologist sitting at a rig is seeing, which is, this is a fresh slice of tissue under the microscope, and you can see a bunch of cells, but the problem is that there is really no way to tell whether this cell is an MSN that belongs to, the direct pathway expressing dopamine D1 receptor, or to the indirect pathway expressing dopamine D2 receptor. And to really differentiate between the two, then we turn on the light, and you can see on the right that some cells really are fluorescent while others are not. And I'm just blowing up this small section of this picture, and you can see these two cells. Of the two cells shown on the bottom right of each picture, only one is fluorescent. So this is how we can determine whether the cell we're recording is expressing dopamine D1 or D2 receptor. So now that we have really somewhat overcome these really technical issues, how do we measure the strength of synaptic transmission in the nucleus accumbens? Well, we have two ways. Either we can evoke the synaptic transmission, or we can record its spontaneous activity. Again, here for the electrophysiologists, I apologize, it's very basic, very simple, but for those of you who don't have an background in electrophysiology, I'm going to spend a few minutes to make sure that you understand what we are doing. So what we focused on is really on the spontaneous excitatory postsynaptic potential. If you have questions about the evoked synaptic response, I can answer questions at the end. So this is really a typical trace. Every downward deflection is actually glutamate binding to the AMPA receptor. So from a typical trace like that, we can extract two kind of information. The first one is the interval between each event. And from that, we can really make a graph that is a cumulative distribution of these intervals. And this really will allow us to really measure the frequency of these events. The second information we can collect from this kind of traces is the amplitude of the events themselves. And we also can make a graph that will really tell us about the distribution of the amplitude. And now that I explained what we measure, how we measure it, and exactly the kind of information we can extract from these traces, let me really tell you very briefly about the protocol that we use. So typically it's very simple. We record the spontaneous activity for 4 minutes. Then we stimulate with light the interneurons. Here we use 470 nanometer because that's the wavelength at which channelrhodopsin is sensitive. And the protocol is, again, fairly simple. It's a series of 5 consecutive very brief pulse stimulation that are separated by 20 seconds, and we repeat this cycle for 2 minutes. And once this is done, then we record the same activity for 4 minutes post-stimulation. So for all the graphs you're going to see now, you will see a curve that is pre and one that is post. So when there is you will see a shift to the right, then this will indicate that there is a decrease of the frequency. And for the amplitude, a similar shift to the right would indicate an increase of the amplitude. So, but before we can do that, we had to make absolutely sure that the light stimulation in and of itself would not really create an issue, a bias. So what we did was to use to record the spontaneous activity, this time in the chat GFP mice. So there is really no chirrhodopsin expressed in the cholinergic interneurons in these mice, just GFP. And what you see is that when we, stimulate these neurons or the tissue, well, there is absolutely no change of neither the frequency nor the amplitude. You can see the white curve and the red curves are perfectly superimposed. So what happens to the activity, the spontaneous activity in medium spiny neurons expressing dopamine D1 receptors? Well, you can see here, even if you are not really electrophysiologist, it's pretty clear that one obvious change is the frequency. And this is a typical recording, but this is confirmed when we pull 10 recordings together. You can see now there is really a right shift of the curve after stimulation of the cholinergic interneurons. Interestingly, when we looked at the amplitude, we saw no difference at all. For D2, with D2, we saw exactly the same thing. We saw a right shift for the frequency. This is the graph on the left, and no shift at all for the graph on the right for the amplitude. So now let's go back to our little cartoon. We know that cholinergic interneurons, when they are stimulated, they will really impair the release of glutamate. But the question that arises is, what is the mechanism? Is it an effect through the release of dopamine, or is it a direct effect? So to answer these questions, we did a little bit of pharmacology using very specific blockers for dopamine receptors and cholinergic receptors. So let's start with the D1 MSN. On the first graph on the left is really what I just showed you in the previous slide. Now, when we put when we superfuse, when we bathe the tissue with mecamelamine and atropine, a specific antagonist of acetylcholine receptors, we can completely block the shift to the right. And when we do the same experiment, this time with dopamine receptor antagonist, supirid and SCH23-390, we get exactly the same result. So not shown here, not shown here, not shown here is the D2. What happened with the D2 for the sake of time? We still see a shift for D2 that we don't see here. So this really, the result of this experiment suggests that when we stimulate cholinergic interneurons, we induce the release of dopamine. But we had to demonstrate that this was actually happening in the slides. So to do that, we turned to a technique that is called the fast scan cycling voltammetry. I'm not going to get into the details of this technique. It's just a carbon fiber that we stick in the tissue, and we can measure the release of dopamine following the stimulation of the cholinergic interneurons. And indeed, when we do that, we found that there was a really nice release of dopamine. So yes, when we are absolutely confident to say that when we optogenetically stimulate the cholinergic interneuron, we induce a release of dopamine. So those are really the questions we had at the beginning. Do cholinergic interneurons modulate glutamatergic synaptic transmission, and what are the cellular mechanisms? Well, what we can say with a high degree of certainty now is that when it comes to spontaneous release, the stimulation of cholinergic interneurons do inhibit the release of glutamate, and this effect is actually not direct, it's totally indirect, is mediated through the release of dopamine. And this is true for D1 medium spine neurons. Now, for D2 medium spine neurons, the situation is completely different. Now, the stimulation of cholinergic interneurons also inhibit the release of glutamate, but this effect is very direct. So here is already a difference between dopamine D1 and dopamine D2 expressing medium spine neurons in terms of the influence of cholinergic interneurons on the regulation of synaptic transmission into nucleus accumbens. But the real question that really we are after is really to understand what are the effects of binge alcohol drinking on the glutamate release. Well, here I'm showing again this kind of graphs where we really focus on the frequency, and on the left, this is really what I showed you previously. When we stimulate the cholinergic interneuron, there is a right shift indicating there is a decrease of the frequency of release of glutamate. And what is interesting here is that when we repeat the same experiments, but this time in mice that have been drinking alcohol for 2 to 3 weeks, this shift is completely blocked. And the other interesting thing is that when we do the exact same experiment in D2 MSN, well, we could not block the right shift. So again, a difference in terms of how the system responds to binge drinking. So here is the situation. On the left, dopamine D1 MSN. On the right, dopamine D2 MSN. On the left, we have a very indirect effect, inhibition of the release of glutamate that is mediated by the release of dopamine. That's for D1, and for D2, a direct effect. When we let the animals drink for a couple of weeks, this effect is completely blocked, whereas in D2 MSN, apparently alcohol has absolutely no effect. So some of you may be behaviorists, and I'm sure you're curious about really whether we tested the effect of cholinergic interneuron on drinking. And we did. Here is our really quick setup. We put animals in a box. They are, as you can see, hooked up to optical fiber and connected to a cannula that is implanted in the brain. And the animals are allowed to drink ad libitum as much as they want. And each time they touch the spout to drink, we get a signal, and we can count the number of times they lick. And here, I'm not going to, for the sake of time, I'm not going to go too much into details, just to say that really focus on this result. And what we found is that when we stimulate the cholinergic interneurons, those mice are the channel rhodopsin mice. There is really an increase of drinking. So it appears that, again, the cholinergic interneurons, and probably through the mechanism that I just showed you, is able to control the amount of alcohol that animals drink. We are repeating this experiment, so I'll come back in 2 months and I can really tell you more about it. Okay, so this is really the second project. It's a little bit different. It's called the integration of emotional and cognitive information by MSN in naive and binge alcohol drinking mice. So, over the past 20, 30 years, a number of theories have been really proposed to explain addiction. I mean, some of them really address the different aspects of alcohol addiction: withdrawal, dependence, stress, whatnot. And, I've listed here probably the main 3 theories: the anhedonia, the aberrant learning, incentive sensitization, and the people who came up with this hypothesis or theories. But the one that really kind of, struck my interest is really, it's called the decision-making and loss of inhibitory control. And this was proposed by Antoine Becard in 2005. So it's not really a new theory, but it hasn't really been tested. And why does it really matter? Well, because when, I said binge alcohol drinking really affects really mostly late adolescents and young adults, when they are really exposed to a situation where they can really binge drink, of course, they are thinking about their parents, who told them, well, don't really touch alcohol, it's bad for you. At the same time, they are in an environment that's, with kids, friends that who say, well, let's have a really good time. So they really, they have to deal with very, very different kind of information. One is the appeal of being part of a group, do like your friends do. And on the other hand, listen to your parents, they know maybe better. So there is really something that the adolescent has to really juggle with. And this theory, the Becerra theory, really is based on the decision-making is under the control of two opposite systems. One is the executive system, originating in the prefrontal cortex, and the other is in the impulsive system from the basolateral amygdala. And that actually the decision to drink, or the consequences of drinking, results from the executive function being overpowered by impulses. And for that to really, the problem with this, the problem with this theory is that it's really entirely based on brain imaging and on testing with patients. And there is absolutely no cellular correlates to this theory. So this is really what we try to really to address, this gap between this theory and the cellular substrate for this theory. So how do accumbens medium spiny neurons integrate competing and executive and emotional information? So the hypothesis is that we came up with is that in naive animals, there is a reciprocal control that we call gating between the cortical and the amygdala inputs in the nucleus accumbens. And this control is really altered after binge drinking. So how do we achieve that? How do we really measure that? Well, here, so what we do is that we go back to optogenetic, because that's a tool that's actually the only tool to really do what we're trying to do. And so the experiment is fairly simple. What we do is that we inject in the cortex, we inject a viral vector that contains, the DNA for the channel rhodopsin, and in the same animal, we inject directly into the basolateral the same viral vector, AAV2, but this time with the DNA coding for a different opsin. It's called crimson. The channel rhodopsin, as I said earlier, is excited at 470 nanometer. In contrast, crimson is excited at 600 nanometer. So we do this double injection, then we wait, and 3 weeks later, we perform the recording. So what happens is that we cut the brain in slices. We put the slice under the microscope, and we patch the medium spine neurons. And when we turn on the blue light, this is really what happened. We can evoke a synaptic response, and in the same recording, in the same cell, we can, with some delay, we can turn on the red light, and this time we excite crimson, and then we get another synaptic response. So this is really the setup. We can then with the delay that we choose, we can stimulate first the cortical inputs followed by the BLA, or do exactly the reverse, start with the stimulation of the BLA inputs and then stimulate the cortical afferents. So, do cortical inputs control the strength of the amygdala synaptic transmission? So let's start with the bottom trace here, which is, with the P1. When we stimulate the amygdala, we can really evoke a very nice synaptic response. And when we do so, but following the stimulation of the cortical inputs with a delay of 165 milliseconds, you can see that, the stimulation of the cortical input has pretty much no impact on the strength of the BLA synaptic transmission. However, when we shorten the delay between the cortical and the BLA stimulation, you can see there is a very nice time-dependent inhibition of the BLA synaptic transmission, to the point when the BLA is stimulated 40 milliseconds following the stimulation of the cortical input, the synaptic transmission of the BLA pathway is completely abolished. And this is shown here in this graph, where you can see at 40 milliseconds, the inhibition is nearly total. It's around 92-93%. But as the delay between the two stimulation increases, then the synaptic response of the BLA pathway recovers really nicely. Now, if we reverse the stimulation order, we start with the BLA followed by the PFC. Well, in some cases, in the panel C, for instance, you can see that the stimulation of the BLA prior to stimulating the cortical inputs with an interval of 40 milliseconds pretty much totally again inhibit the cortical synaptic transmission. However, in some cells, and this is shown in the panel D, this inhibition is very weak. And this is really what we have. At 40 milliseconds, we have a great variety of responses. In some cells, the stimulation of the BLA inputs will totally inhibit the synaptic transmission of the cortical pathway, while in other cells, it will fail totally. So now you can really see the contrast between the two situations. Well, quickly here, what I want to show here is that this gating phenomenon, this inhibition by one pathway over the other, is extremely sensitive. Even when we decrease the amplitude of the cortical input, from the strength of the synaptic transmission of the cortical input, you can see that it still really inhibits totally the synaptic transmission at the amygdala pathway. But here, again, the real question is really what happens when the animals drink alcohol for a couple of weeks. Well, what we found is that actually it does have a substantial effect. So let me really show you actually side by side. It might be easier. This is really on the left what we got in naive ethanol naive mice. On the right as after binge drinking, and you can see that especially at 125 milliseconds in naive animals, the BLA was totally unable to really inhibit the synaptic transmission on the cortical pathway. Now, after binge drinking, the situation is really different. Now we have now the BLA in some situation, in some cells, is able to almost nearly completely block the synaptic transmission at the PFC pathway. But in those cells, it's not so. So there is really a mix. And this is true also for an interval between the two stimulation of 115 milliseconds. For the PFC BLA, well, we didn't see really much of a difference, to be honest. As you can see here, at 125 milliseconds, the BLA synaptic transmission, the strength of the BLA synaptic transmission has pretty much recovered in binge drinking as animal as it did in ethanol naive mice. So, what is the conclusion of this this part of the talk? Well, it shows that really there is a reciprocal control between the cortical and the BLA pathways. That the synaptic gating is presynaptic. I didn't show the data again for the sake of time. Again, didn't show the pharmacology. We did because again, time constraint. But what we found is that the gating is absolutely not affected by mGlu receptors antagonist, dopamine antagonist, cannabinoid receptor antagonist. So we do believe that the gating is probably mediated by AMPA receptors. We don't rule out the possibility that GABA receptors might be involved, but this is something that we'll test in the coming weeks and months. In naive mice, the inhibition of BLA input by the cortical pathway is stronger than the reverse. And finally, in binge drinking animal, this really balance between the two inputs is really quite profoundly altered. Now, in binge drinking animals, the ability of the amygdala pathway to inhibit the transmission of executive information coming from the cortical area is really markedly strengthened compared to ethanol naive mice. Okay, so now we're going to change gear. I believe we're getting close to the time that was allotted to me. It's really a different project that we initiated just a few months ago that is quite different in nature, but I would like to really share that with you as well. So this is about what we call engrams or neuronal ensembles. The nucleus accumbens, of course, even for a layperson, I suppose, when we talk about the reward pathway, the VTA accumbens pathway, everybody knows that the nucleus accumbens, its primary role maybe, is really to encode reward. Reward to drugs of abuse, but reward to everything. However, if you look at the literature, you will see that it's not a one-trick pony. It's also involved in other functions like sleep and depression, and the list goes on. So the question that we are asking here is whether the neurons that encode these different functions, addiction, sleep, and depression, are completely different. Do they somewhat overlap, or do they totally overlap? So here is the slice. You can see the neurons, you can see the recalling pipette. And as electrophysiologists, when we patch these neurons, we can absolutely tell you whether it's a medium spine neurons, whether it's a GABAergic interneurons based on the biophysical properties. But what we cannot tell you for sure is that whether the neuron that, this person is really recalling is involved really in addiction, sleep, depression, feeding, whatnot. And so what do we care? I mean, you're going to ask me, why do we care about that? Well, we care because hopefully one day, if we want to treat with some degree of efficacy addiction, then if indeed these engrams, these cells that encode these different functions, are totally distinct, then that we might be able to say, well, let's go after the addiction cells and leave alone the cells that are responsible for sleep, depression, feeding, and so on. Because the problem with drugs, as they work today, is that it's really a blanket approach. They will block dopamine, synaptic transmission everywhere in the brain, or will alter the release of 5-HT everywhere in the brain. So it's absolutely not targeted. And as we know, most drugs have really serious side effects, and their efficacy for, most of them is really not that great. And maybe the problem is that there is really no specific approach. So if we can really determine whether the neurons, these engrams, are really distinct or not, then that could be the first step towards really developing a very specific approach that would really target, again, a very specific aspect, addiction, sleep, or depression, without really affecting the other functions. So how do we do that? Well, we use an mediatory gene that is called CFOS, that is known to be responsible for long-term potentiation and memory formation. So what we know, and this has been really shown for, almost 30 years, more than 30 years, we know that when animals really drink, then there is a number of cells that are excited and CFOS is expressed. And here I'm showing you a paper by Bachol that was published in 1990, where the binge drinking really excites a number of cells, and you can see the staining in brown of the cells expressing CFOS. And this is something that we have shown again, in our lab. Here on the left, all the cells are labeled with neuron-specific marker, NUN. When we inject saline, well, nothing really happens. And however, when we inject IP alcohol, you can see that there are a number of cells that really are excited by the injection of alcohol. What is interesting is that when you look at the number of cells, really, it's very low. Here it's around 5%. So you may think that this is really unique to alcohol. Actually, not at all. I mean, the first people who really developed this approach, it goes back to the mid-2000 years, 2007 here in the lab of Mark Mefford. He looked at the expression of CFOs in mice that were trained, for fear conditioning, which is a very, very strong behavioral paradigm. And he found that in the BLA, only 3 to 5% of the cells were labeled. So the fact that, very few cells are labeled by any given, stimulation, it's something that really seems to be true for any stimulation. So how do we do that? Well, there are a number of transgenic mice that we could use to really capture the expression of CFOs in cells that have been activated by alcohol. The one we settled on is called CANE. It's capturing and manipulating activated neuronal ensemble. And it's a mouse that was developed by Dr. Wang, that initially was at Duke, now is at MIT. So this mouse on the face of it is really fairly straightforward. The CFOs promoter really drives the expression of TVA. So what is TVA? TVA is an avian protein that is not found, obviously, in mouse. So each time, really, the neurons are really excited, be by alcohol or anything else, then the CFOs expression is going to drive the expression of TVA. So why is that really important here? Because when TVA is expressed, so you can see here, for instance, there's a bunch of neurons. The animal is binge drinking. A fraction of these neurons really will express CFOs, which will drive the expression of this protein TVA. And TVA will be expressed in the membrane of these CFOs positive cells. So now if we inject a couple of hours after the experiments a virus, lentivirus, that is coated with the ENVA protein, well, this virus will bind very specifically to the CFOs TVA expressing cells. And if we co-inject with this lentivirus another virus, like an AV, that has a DNA that codes for CRE, for GFP, RFP, or channel rhodopsin, or any actually kind of opsin, then we can express very selectively channel rhodopsin, for instance, in the CFOs positive cells. And so what is the idea at the end? The idea is really using this approach to not only functionally characterize the cells that are CFOs positive, to perform single-cell RNA sequencing, to manipulate engram excitability, and this is really ultimately what we want to do, to see whether if we turn on or off these neurons that have been very selectively activated by binge drinking, can we really change the drinking behavior. And also, but this is really further down the road, compare addiction and stress engram in the same and different mice. Okay, so the conclusion. What we can say about the physiology and the synaptic transmission in the nucleus accumbens, the relation with cholinergic interneurons, what I showed you is that cholinergic interneurons in D1 medium spiny neurons really inhibit the release of glutamate through the release of dopamine. And in D2 MSN, this effect is direct. When we also showed you that the cortical and the BLA inputs really have a crosstalk, they inhibit each other. And then when we let animals drink, when they binge drink, the effect of alcohol is on the indirect effect and also on the crosstalk. So here we have the perfect example of a kind of multi-pronged approach in terms of the strategies that alcohol is using to modulate the synaptic transmission in medium spiny neurons and ultimately to really change their behavior. So of course, this would not be possible without the contribution of my collaborators. Genia Kopakova, she is a graduate student. She's finishing. Now she will probably defend in September or October. She was in charge of the cholinergic interneuron project. Timili just came on board. He's going to work on the synaptic gating story. And finally, Dr. Pablo Jimenez Gomez is working on the neuronal engram. And I would like also to thank Dr. Wang for sharing her mice and her vector, viral vector, and Dr. Thompson here in the department will be doing a single-cell RNA-seq. Last but not least, I also would like to thank the National Institute of Alcohol Abuse and Alcoholism for the financial support. And finally, I would like to thank you all for listening to this presentation, and I would be glad to take some questions. Thank you.
Fantastic. Well, Dr. Martin, we'd like to thank you for a really great presentation. All right, so moving on to the Q&A, I'm just going to add Gilles back onto the audio line. So first question today comes from Shin, who's asked, Gilles, are there any significant alcohol metabolism involved in enhanced alcohol intake animals?
Well, that's really, I'm not sure what the right answer is about alcohol metabolism per se. I know some people really do process really alcohol faster than others, but in this particular situation, it's unlikely that this really plays a major role, simply because, we're working with mice line. They are all the same background, C57, black 6. So I cannot really totally rule out that there might be some differences between mice, but it's really quite unlikely.
Got it. Perfect. Thanks so much, Gilles. Yeah, a few more questions here coming in. Gilles, what type of binge drinking protocol have you carried out? So what's your actual method for getting the rodent models to ingest the alcohol?
It's a model that's been around for quite a while now, that has been developed by John Crabb at the University of Oregon. And it's really pretty straightforward. We allow the animals to drink for 2 hours daily in the dark. And the key word here is in the dark, because otherwise mice really don't drink readily, unlike rats. So every day we use a reverse dark light cycle. And every day we allow them to drink for 2 hours. And we repeat this drinking session for 5 consecutive days. We give them a 2-day break, and we repeat the same protocol the following week, and this for 2 or 3 weeks.
Excellent. Thank you. Next question here comes from Julia, who's asked, how do you foresee how your work will inform the development of pharmacotherapies or any other kinds of therapies for alcohol use disorder?
Well, that's an interesting question, actually, and this is something I've been thinking about quite a lot. And this really, I think about that in the context of the last project that we initiated a few months back about the engrams, on the last project that I showed in this presentation. Because the whole point of this project, as I tried to explain, was really to identify anything in terms of the properties of the neurons that are specifically recruited during binge drinking that may not be recruited during depression or during feeding or stress. So if we can really identify this neuronal population with a high degree of precision and see what is unique about it, maybe we can devise pharmacological approach that will very specifically target this subpopulation of accumbens neurons, either by turning them off or doing exactly the opposite by exciting this engram while leaving the other neuronal population in the accumbens totally intact. But at this point, this is still very early. So to give you a precise answer, it's maybe in a few years, but now it's all I can say about it.
Interesting. Well, great answer there. Here's a question from Greg, who's asked, in the experiment where the cholinergic interneurons were excited and the animals increased their consumption of alcohol, was the alcohol cued in any way, or was it just free access?
No, it was not cued at all. The animals are really allowed to really drink as much as they want. As I showed in one of the slides, they have total free access to alcohol for 2 hours. And I didn't show this piece of data, but we measured exactly, how much they drink over the 2-hour time period. And what we found is that they typically drink the bulk of alcohol during the first hour. And after that, it tapers off during the second hour of the session.
Excellent. Okay. All right, maybe in the interest of time, we'll just have one last question here. Swalin has asked, have you investigated or observed differences in cholinergic interneuron activity or in the reciprocal connections between BLA and PFC between females and males?
Great question. We are doing that right now. So we don't have the answer. We have some ideas of what's going on, but it's really too early to tell which way it's going to go. So I prefer to really to wait until we get all the data to answer that question. So maybe next time.
Excellent. Yeah, definitely maybe next time. Well, Gilles, thanks so much again for the really fantastic presentation and for your insights in the Q&A today. It's been a real pleasure having you with us.
You're welcome. Likewise.
Excellent. And also a big thanks to the audience for joining us today. And last but certainly not least, I'd like to give a big thank you to Harvard Bioscience for sponsoring this event to make it possible. So in closing, thanks everyone for taking part in this Inside Scientific webinar, and we're looking forward to having you with us next time. Have a great day, everyone.